Why am I always shocked when it shows up? I don't know. Let's see your faces this morning, people. Let's just say hello. There we are. Matt Gremlin, Noemi Alonso, Dante Quinones. Dante, are you like bald? Because you always have like a hood on or something. Are you like bald back here?
Nah,
Oh, score. The man bun. I love it. I love it. I love it. Oh, Kaylee Quackenbush got up late and didn't get a shower, so she's not going to show us our face. That's okay. That's okay. I'm down with it. Oh, there she is. She did get a shower. Looking good, Kaylee. 1, 2, 3, 4, 5, 6. Only 6 people. Let's wait a little bit and see who all else shows up. My lecture's a little short today, I think, although like 15 minutes ago, I threw a whole nother section in just because I thought, oh my God, this is gonna be half an hour. So I'm not sure how long it's gonna be today, but a lot of it's review. Let's see. Celine, have you had cell biology? No?
Yeah, a while ago with you.
A while. How long ago was a while ago?
A long time ago that I don't remember anything.
Good. That's what I want to hear. Ariana just had it last spring, but and she hasn't forgotten anything because she doesn't. Noemi, have you had cell biology? No? Okay. Kaylee, were you in cell biology last semester? Dante had cell. Matt had cell. Mike West had cell. Oh, Mike West is here. Hello, Mike. Did you cut that damn hair yet? Let's see your face. Mike West, shortest name in the class. Nicole's here. Yay, Nicole. What do we got? We got 8 people. When you're in this class, I think 14 or something. Should we go ahead and start? I guess we're not in a hurry. We don't have that much to do. All right, why don't we go ahead and get started? You can turn your video off if you want. It's up to you. I'm just not going to make you keep it on, but you can leave it on if you want. I'm going to open my chat, so if you need to ask a question, we're there. And we'll start today by doing a little bit of review. Can you hear this? The geese are starting to flock up out here. It's that time of year. Okay. Okay, so I'm gonna start by introducing you to this guy, Robert Furchgott. I'm sure you've never heard of him. Dapper looking fellow. Not really. Looks like a grumpy old man. But he discovered a couple of really important things that we're going to talk about now. One is that, well, spare receptors is kind of important, not extremely important, but the really important thing he learned about was nitric oxide, which we'll talk about probably on Thursday. I think he got the Nobel Prize for that, so pretty important discovery. And I always like to, buck up the troops in terms of pharmacology. He was a really good pharmacologist. Okay, so Robert Furchgott. So before we get to his discovery of spare receptors, let's kind of review what we talked about last week. This is about potency and efficacy. So your potency is your your ED50. What concentration of drug does it take to get 50% of the maximal response of that drug? And so that's potency. And potency and efficacy are 2 different things. You can have a potent drug that produces a little effect. You can have a potent drug that produces a lot of effect, on and on. And if you've got a drug like C, doesn't have any efficacy, you have to, through other experiments, show that it is actually binding to that receptor. But if it's binding to that receptor and has no efficacy, that makes it an antagonist. Try to pull my chat window up. Oh well. Celine, I can see your face, so if somebody chats and I don't respond, just give me a little wave. Tell me I need to check that. Okay. Okay, so potency and efficacy. So here's the effect that a competitive antagonist has on the dose response of an agonist. So the competitive antagonist is binding to the same receptor and not activating it. So they're really physically competing for each other. Whichever one has a higher affinity and is a higher concentration will win out. So you can always beat off that competitive antagonist by going to higher concentration. But since they're competing, it seems to change the potency of the agonist. So bottom line being, when you add a competitive antagonist, the ED50 shifts to the right, which means the apparent potency of the agonist decreases, but the maximal effect stays the same because those receptors are all there. You go to high enough concentration of agonist, you'll still get a full effect. Non-competitive is different. This is when it binds to a different site on the receptor than the agonist, so they're not competing for the same site anymore, but it turns the receptor off so that the agonist can no longer use that receptor to produce an effect. So when you add a non-competitive or irreversible antagonist, what happens is you decrease the efficacy of the agonist, but not the ED50. So the ED50 stays the same, but the effect decreases. This, by the way, phenoxybenzamine, that's why I got distracted there. I'm looking at it because phenoxybenzamine is a classic irreversible antagonist. It binds to the receptor covalently, so it basically just destroys that receptor. They're gone now. And we'll see that. I only know one disease where that's used, so it's not very commonly used, but it's good in research to figure things out. Okay, effect of the partial agonist on the full. Now this is the most complicated of our dose response curves. So to understand what's going on here, there's your dose response of the full agonist. There's your dose response of the partial agonist. So what the third experiment here is we put like this concentration of partial agonist in. So you already have this much effect before you start adding your full agonist on top of it. So there's the effect of the partial agonist. Now for the full agonist full agonist has to compete against that partial agonist to get in there and then fully activate that receptor. So it's similar to, at this point, the interaction between the full and partial is similar to the interaction between full and a competitive antagonist. So you have to go to higher concentration of full to get the full effect. Okay, so the full antagonist has to compete off the partial to get full effect. And that gives you that rightward shift or the apparent decrease in potency. And what's important about this is, I think the most important drug for this, and again, it's not this class because it's neuropharmacology, but the best example of this is oh God, what the hell's the name of it? Buprenorphine, which is used for heroin addicts because, if you give them an antagonist, they'll go into withdrawal. So you don't want to, and if they stop taking the full agonist, heroin, they'll go into withdrawal. So you give them the partial agonist, so they're not going into withdrawal, but they're not getting high either. So it's kind of maintaining them at a certain level. Okay, I'm trying to think if there's a cardiovascular partial agonist. I'm sure there are, but I can't think of them right now. Okay, so that was last Thursday. Any questions about that? Any comments? Any discussion vis-a-vis that? Okay, good. Decrease apparent potency. Okay. So the next thing to talk about is spare receptors. So if we back up, let me show you this dose response curve again. I'll show you this dapper fellow again. So here, when we look at this, what we're assuming here is that you get full effect with norepinephrine, when you activate all those receptors. And as you decrease the number of receptors, you decrease the effect. Makes sense, right? The problem with that is the assumption that you need to activate all the receptors to get 100% effect. And what Furchtgott figured out was that's not necessarily true. What he realized was that there are more receptors present than needed. So you get 100% effect with less than 100% of the receptors occupied or activated. And you can see this when you compare the affinity of the drug to the ED50. Now, if every receptor had to be activated to get full effect, then you would have to have the occupancy at B max, every receptor occupied in order to get full effect. But with most systems, that's not what we see. And Furchtgott used a really good model for this because there's so many spare receptors here. This is the norepinephrine and a rabbit aorta. It was aorta. It might have been a different vessel, but I'm pretty sure it was rabbit aorta. If you do the binding experiment with your binding isotherm, this is what you get, and you find out that the KD is right about here, right, half maximal. But if you do the pharmacology and you do contraction studies with the rabbit aorta, your ED50 is way down here. And you do the math on it, you find out that right here, in terms of your binding, only 6% of the receptors are bound with norepinephrine, and you're getting 100% of effect, which tells you that 94% of those receptors are extra. They're spare receptors. And why is that? That's my next thing. So why is that? Well, if you go back to our binding isotherm, we said ligand plus receptor goes to ligand receptor complex, right? And we're always talking about changing ligand concentration to increase the bound receptor. Well, the cell can do something like that too, where if it needs to be more sensitive to the ligand, it can increase its receptors. So if you increase the number of receptors, that also shifts it over to the right, and you get more. You get. Oh, actually, I'm sorry. Shifts the potency of the ligand to the left. So the ligand appears more, or becomes more potent when there are more receptors there. So we'll talk about receptor regulation probably in the next class, but basically the more you stimulate a tissue, the more it removes those receptors. The less you stimulate a tissue, the more it puts up there. So something like the rabbit aorta has got a lot of receptors there, and so it's very sensitive to norepinephrine, probably because it doesn't see much. It's not often your aorta will be contracted for some reason. So it's got a lot of extra receptors there, spare receptors. Norbert, can you close the door for me? Oh, unless the dog gets up. Okay, so that is spare receptors. Now, how do you tell if you've got spare receptors there? Well, thanks for asking. We'll go back to this, okay? So with no spare receptors, as soon as you start destroying receptors, you decrease the effect of the agonist, right? So take receptors out of the equation, you get less effect with that agonist. If you've got spare receptors, though, as you decrease the number of spare receptors, you shift the apparent potency of the agonist to the right. Okay, you've got fewer. You remember the on rate is based on the concentration of both receptor and ligand. If you start decreasing ligand receptors, you have to go to higher concentration of ligand to get the same amount of LR. So this is IA in irreversible antagonist. As you increase the irreversible antagonist from 0 to 10, whatever, nanomolar, whatever, you shift the apparent potency of the agonist to the right. You increase to 30, force it to the right as well. Now, at some point, you're going to destroy so many receptors with that irreversible antagonist that you can no longer get full effect. So now we're back to the point where there are no spare receptors. And increasing irreversible antagonist then has the effect you expect with the irreversible antagonist. You destroy the response by destroying the receptors. So the key point being here, when you've got spare receptors, an irreversible antagonist will shift the curve to the right, decreasing the apparent potency until there are no more spare receptors. And at that point, you will, the KD will stay the same, but the efficacy will decrease. Okay? Let me stop the share for a second. Oh, Mike, give us a little man bun or something for Christ's sake. You look like a hippie.
Should I?
No.
I'll do it. I have one right here.
No, I'm being a jerk.
Yeah, 1 second.
I'm being a jerk.
No, it'll be funny. Sorry, I was having trouble connecting. I just got a new laptop and it's acting all funky.
Oh, that's fine. It's no biggie. And I always post these lectures later if you miss it, so it's not a big deal. Okay, let me see your faces, guy. I have to ask you a question. Eunice. Okay, so did all this make sense? Yes or no? Spare receptor stuff? All good? Okay, good. Mike's in. Okay. All right, so let's move on then. Let's see, we've already killed 15 minutes of the class. I had hoped that would take 30, but on we go. Like I say, it's hard. You know, if I saw you guys in person, I could BS my way through an hour and 15 minutes like nothing. But here we are online, and my BSing capabilities are a lot less. All right, spare receptors. And now we're going to go into receptor pharmacology. So all that stuff is classic pharmacology stuff. A lot of that stuff was worked out before anybody knew about receptors, knew what they were, or what they did, or how they operated. I mean, it was all pretty empirical. You know, we know this drug has an effect. Well, let's see how we can figure that effect out. We go to different concentrations, we do dose response, blah, blah, blah. But no one knew what happened when the receptor, after the receptor was activated. That kind of information started really in the 80s, a little bit before I hit grad school. People started figuring out about G protein coupled receptors around then, and they were the big hot topic. So that's what we're going to talk about now is G protein coupled receptors and how they produce effects. So that's receptor pharmacology rather than the classical pharmacology. So the binding of ligand activates the receptor. Activation means a change in conformation. New domains of the receptor are exposed to cytoplasm. They start a chain reaction in cytoplasm. Okay, so basically we talked in the first class about how you've got the receptor. When the ligand finds its way down in there, it binds, and it's binding because those little, what do you call them? The functional groups on the drugs interact with the functional groups on the receptor, on the amino acids of the receptor. So that causes the receptor to pull together to grab that ligand. And when it does, that movement, that conformational change translates to the whole rest of the protein. So new domains get exposed. And that's what activation of the receptor is. When the ligand binds, the conformation of the receptor changes in that little spot, and that little spot translates to the whole rest of the protein, and then new things get exposed. Once those some new domain gets exposed, it then interacts with some other protein that's there, and that gets activated in the same kind of way, and then activates something else, and so it's all a chain reaction, starting with the ligand binding to the receptor. Three main pathways we want to talk about here. If you remember cell biology, I talked about probably 10 or 15. I don't even know. It's just like on and on and on in that stupid class. It's funny. I remember years ago, do you guys remember TGF-beta receptors if you're in cell biology class? TGF-beta receptors bind together, then they activate the SMADs. And SMAD, some mothers against decaplegic or something like that. That's what it really stands for. But one of my students in cell biology back then called it so many asinine diagrams. That was the section on receptor mechanisms. But anyway, just three we're going to worry about here. And hopefully you know them all already, but this gives us a chance to review. So number one, cyclic AMP. This will be really important. This is our almost exclusively sympathetic second messenger. So the sympathetic nervous system activates production of cyclic AMP, not always, but mostly, and we get results with that. Another aspect of the cyclic AMP is actually inhibiting cyclic AMP production, which is another receptor mechanism, but very similar. Then we've got our IP3 calcium, protein kinase C pathway. And I say protein kinase C and others because it'll activate protein kinase C, but it'll activate other kinases as well. And they will produce the effect in the end. And then nitric oxide, which causes the production of cyclic AMP. And then you've got a different kinase there, protein kinase G. And we'll see why that's really important in arterial smooth muscle, especially. It has the same overall effect as cyclic AMP, but it's got different targets. So it's even more effective at relaxing smooth muscle. So all of these things are related to G protein receptors. So these are all being activated by G proteins in some way. Here are the G protein receptors. This is kind of the basic snake diagram, we call it. So the receptor doesn't really look like this, but if you splay it all out and look at it, you've got 7 transmembrane regions. which is why they're sometimes called 7 transmembrane receptors. And let's see, we've got 1, 2, 3, 4, 5. And then you can actually, by doing site-directed mutagenesis or swapping out different pieces of these receptors, they figured out where actually the ligands were binding and what parts of the protein were important for activating G proteins and such. This drawing now I'm looking at, I'm just looking at it a little more critically, pretty archaic, really. I mean, this is, they're saying the ligand binds here, but it also binds to some amino acids over here. And these loops here actually fold into the protein like this. They're not sticking up like this. They're folded down in. We'll see that in the next picture. But bottom line being the ligand binds out here. And then that causes a translational or a conformational change that exposes this loop here, and this loop here interacts with the G protein. So I'll get that. And then also you've got the regulatory domain. When we talk about proteins being, or receptors being down-regulated, we're talking about phosphorylation at these sites. So you've got these, where are they? But I was going to call them magenta. That's definitely not magenta. That's like a reddish orange here. These reddish orange sites are consensus sites for phosphorylation by the G protein coupled receptor kinase, GRK. We'll talk about that on Thursday. These star sites are phosphorylation consensus sites for protein kinase A phosphorylation. So there's a lot of different sites on the cytoplasmic side of this protein that can be phosphorylated. What was I going to say? Activation domain. So, if I had to make a guess, I'd say there's 150 different G protein coupled receptors. We'll talk about 3 of them and subtypes of those 3. If you go into the brain, there's more that we're not going to be talking about. Where most of these G protein coupled receptors are, though, which is kind of weird, is in the nose and in the mouth, because your taste receptors and your smell receptors are all G protein coupled receptors. And there's a lot of different ones of those, and they all activate G proteins. I met the woman who actually found that out at Harvard Med School. She got the Nobel Prize for it, too. Okay. Okay, so here's a more realistic view of what the receptor looks like. So if you did X-ray crystallography and you saw what the protein actually looks like, it's like this. And down in the middle here is where that ligand binding site is. And, you've seen this before. We showed this the other day. Here's your, this is norepinephrine actually binding to the receptor. Hydrogen bonding, some ionic bonding, and they're not showing it here, but there's also a phenylephrine interacting with the pi electrons here of the drug as well. And you can see here that drug is like way down in. So those, like I was saying, those things that are sticking out are actually down like this, and the drug is interacting with them down there. Okay, G proteins. How many people are tired of hearing about G proteins at this point? Okay, but I learned it in college, so you have to learn it now. Here's your inactive G protein. So the alpha subunit, the G protein is made of 3 subunits, the alpha subunit, and then the beta and gamma subunit. The beta and gamma are always found together, so we kind of call them the beta gamma subunit, but there's 2 separate proteins. But anyway, you've got these 3 proteins, hetero-trimeric, hetero, 3 different, tri, 3 meric unit, hetero-trimeric. You've got the alpha subunit, which has got the G protein binding domain on it. So the alpha subunit can have GDP bound to it, in which it's off, and when it's off, it'll bind to the beta gamma. Now, if you've got a guanine nucleotide release protein, which can also be called a GEF, G protein exchange factor. So you've got that activated receptor, opens up, and you get exposure of that internal domain. That is your GEF or your GNRP. It's going to kind of grab that alpha subunit and reach into it and open it up, and GDP goes floating off, and then it lets go, and now GTP will bind to the alpha subunit. Once GTP binds to the alpha subunit, it goes into a different conformation. It's now the active conformation, and it will bind to other things and activate things and produce effects. And the beta gamma, as far as we know, is just kind of floating around waiting to snatch up the alpha subunit. So this alpha subunit has got a GTPase activity. So it's going to break down GTP into GDP, and when it does, it shuts off. And there are also some proteins there that will activate the GTPase activity. GTPase activating protein. As far as we're concerned, this is not that important. That's more important in your growth factor receptors. So in general, just think of it in terms of you've got your inactive G protein with GDP on it. The activated receptor comes along, causes it to release GDP. It then picks up GTP and becomes active. And eventually it will shut itself off and go back into the inactive form. So as long as you've got activated receptor there, it'll get turned on. So that's why we call this G protein cycling. It'll just keep being turned on, turning itself off, turned on, turning itself off, or over and over again until the ligand goes away, the receptor is no longer active, and when it shuts itself off, it stays off. G proteins. Okay, cyclic AMP. We all know the structure of ATP, I hope anyway. Here's your ribose sugar, there's adenine, and now you've got 3 phosphates attached to it here. This first phosphate is a low energy bond, and then these squiggle bonds are very high energy, so that's 7.5 kilocalories per squiggle there. So this is the energy from this is going to drive the reaction forward when the enzyme's ready. So when you've got your G protein bound to the adenyl cyclase, again, it changes conformation, it opens up, and it accepts ATP. And when ATP comes in, it breaks this bond here and attaches this over to this carbon over here. So now you've got a circle. It's like AMP, but it's got a circle in it. What shall we call it? Cyclic AMP. There we go. So I'm sorry. I've had a lot of coffee this morning and I'm silly. Okay, so cyclic AMP then will go off and activate some kinase, or kinase, protein kinase A. And here's the enzyme that will shut it off, phosphodiesterase. And this phosphodiesterase, you should just assume it's always on, okay? So you've always got phosphodiesterase activity there in the cell. So cyclic AMP will be broken down into AMP by phosphodiesterase. And again, in terms of the cycle and everything, as long as you've got your G protein activated, you're activating adenyl cyclase, you're making cyclic AMP's levels come up. Now you're constantly breaking it down, but you get to a point where you're making it as fast as you're breaking down, you reach that equilibrium, you've got a high level of cyclic AMP. Ligand goes away, G protein shuts off, adenyl cyclase shuts off, phosphodiesterase breaks down all the cyclic AMP, and it decreases in the cell again. So think about it in those kind of dynamic terms, like it's not necessarily 1-to-1 to 1-to-1, but there's all this going on at once, and it's dynamic. Okay, so putting it all together into one thing, and if this is unclear, I would seriously suggest you go to YouTube and look for a video, because there are a gazillion videos on how this works out there. So anyway, here's your, say, beta adrenergic receptor, and Mr. epinephrine comes along and binds to it and activates it. Right now, your G protein is off, your adenyl cyclase is off. Once that epinephrine binds to the beta receptor, it becomes a GNRP, and it causes the alpha subunit to release GDP and pick up GTP. It will then go over, bind to the adenyl cyclase, and make cyclic AMP. And then cyclic AMP will then go and activate protein kinase A. And this, as long as the ligand's there, this will keep cycling, you'll keep making cyclic AMP. But eventually, when this is off, cyclic AMP gets broken down by phosphodiesterase. All right, let me go to my notes because I think there's more detail there. I found a new button. New share. Where am I? Where am I? Where am I? Oh, come on, man. Come on, man. This isn't it. Is it not open? Hang on a second. Technical glitch. Oh, I closed that file. That's why. All right, sorry. Oh, why? But I'm singing stupid Christmas songs this week. Okay. We are Santa's elves, feeling Santa shows. We are Santa's elves. Okay. Spare receptors we talked about already. Okay, cyclic AMP. So as in cell biology, it's important to know like how these steps get turned on and how they turn themselves off. Which ones are amplifying and which ones are stoichiometric. So I guess that amplification versus stoichiometric isn't that important. Just know that this amplifies the signal. When the one molecule of epinephrine binds to a receptor, you get a lot of molecules of cyclic AMP being made. Okay, so just to go through the series here, hormone epinephrine binds to its receptor. The active receptor interacts with the G protein GS. So that's a little bit of detail here that wasn't in the PowerPoint. So you've got different isoforms of the alpha subunit, and the particular one this receptor will activate is alpha S. So we call the whole thing GS. The alpha subunit is alpha S. Okay. Before interacting with the receptor, the alpha S is GDP bound. The receptor causes it to drop its GDP and then pick up GTP. Alpha subunit diffuses across the inside of the membrane. Remember, this is membrane bound, so it's really operating in 2 dimensions rather than 3, like you might think, because it's it's only across the membrane here. So it diffuses across the membrane, binds to adenyl cyclase, activates adenyl cyclase, and starts producing cyclic AMP. Cyclic AMP then diffuses into the cell and finds protein kinase A. So this is something that I didn't talk about. Let's do that real quick. Share. So cyclic AMP is going to find this protein kinase A. So protein kinase A is a heterotetramer, I guess. It's got 2 catalytic subunits and 2 regulatory subunits. So the regulatory subunits, when cyclic AMP isn't around, are binding to the active sites here of the catalytic subunit. Cyclic AMP binds to those, and when cyclic AMP binds, the regulatory subunits come off, and your catalytic subunits are released. Experimentally, by the way, if you wanted to see what cyclic AMP does, you would just inject the catalytic subunit without the regulatory subunit. Fun fact. Okay, so once your catalytic subunit is released, it's going to go off and phosphorylate proteins. But again, not just any random protein out there. It's only going to phosphorylate proteins with serines or threonines, and only those serines and threonines that come in a specific consensus sequence. So here's our consensus sequence for phosphorylation. Arginine, anything, and then a serine or threonine, and then something hydrophobic. Okay, so that's our consensus site. If this enzyme comes across anything with, not this site, but has a serine or threonine, it doesn't phosphorylate. It needs to grab all those things together, recognize it, and then phosphorylate it, and then move on. So that's our consensus site. Okay. Okay, so catalytic subunits then phosphorylate target proteins and produce effects. And that'll be kind of the next part when we talk about the physiology of the cardiac cell, the physiology of the smooth muscle cell. What are the targets that get phosphorylated, and how do they produce the effects that are seen with that second messenger? Reversal of the mechanism, also important. I was kind of harping on this all the way through. Hormone comes off the receptor, right? Non-covalent, hops on, hops off. Receptor goes to inactive. Alpha subunit hydrolyzes GTP to GDP, shutting itself off, rebinds with the beta gamma. The alpha subunit came off AC, so AC is turned off. Cyclic AMP is broken down by phosphodiesterase. Its concentration falls in the cell. When it falls, cyclic AMP comes off the regulatory subunits, and then the regulatory subunits bind to the catalytic subunits again, shutting off pKa. Now, all those proteins that phosphorylated are still phosphorylated, so you've got these other enzymes, phosphatases, that are going to take the phosphates off the target proteins and shut them off. So phosphatases are, you should think of in the same way you think of PDE, phosphodiesterase. It's like these are the shut-off enzymes, and they're always on. So to get a response, you have to continually produce the response, and then to shut it off, you just stop making the response, and these enzymes shut everything down. So those phosphatases will go around and take the phosphates off the target proteins, and that'll shut off the effect of the ligand. Okay? We good? Yes, everybody's good? Okay, the other part of cyclic AMP, again, if you start thinking about this in a more dynamic sense, I guess when we teach these things, you always try and simplify it. I had a teacher in high school who said, teaching science is teaching a series of lies. So we tell you a lie that makes sense, okay? And then later, when you get to a higher grade, you're like, yeah, but that's not exactly right. This is closer to right. Oh yeah, but that's not really like, it's like teaching a series of lies. So when we teach this cyclic AMP pathway, it's always like one drug binds to one receptor, activates a G protein, activates HC, activates cyclic A, and it sounds like it's a single line of one molecule doing one molecule. But in a dynamic sense, you've always got a lot of cyclic AMP. You have to have a lot of cyclic AMP being made because it's constantly being broken down. So if you want an effect, you have to constantly make this, and then you're talking about the levels of cyclic AMP going up and down based on ligand interaction with the receptor. And one of the things that we usually don't talk about is baseline levels of these second messengers. We say, there's when you activate it, cyclic AMP gets produced. Well, there's always cyclic AMP being produced at some low basal level. So, and it's probably due to proteins change conformation, right? So the receptors are probably slowly changing to active and inactive form, mostly being in the inactive form, but occasionally going to active form and then back to inactive. So that's a conformational change that happens without the ligand. So when it's doing that, it's also activating alpha S at a very, very low level, which is activating AC at a very, very low level. So even when there's no ligand there, there's some cyclic AMP there, and that's our basal level. Other mechanisms can decrease that basal level, and this is an example of that, inhibition of cyclic AMP. So a hormone like acetylcholine will bind to a muscarinic acetylcholine receptor. We'll talk more about them later, but activating that receptor, and that's going to activate a different G protein, GI, for inhibitory. GI is an alpha I subunit, and again, our beta gamma subunit. I think this is the same beta gamma as with the alpha S, but I'm not 100% on that. I don't think there are other subtypes of the beta gamma, but there's definitely subtypes of the alpha. So this is alpha I, structurally very, very similar to the alpha S, except that when it has GTP, it will bind to adenyl cyclase and inhibit it. How does it inhibit it? Probably competitively with alpha S. So it's going to bind to that site and block alpha S from getting in there. And what this does is decrease the basal level of cyclic AMP. And also if you've got both hormones there, the activator and the inhibitor, you're going to have a lower level of cyclic AMP than if you just had the activator there. So this is what we call physiological antagonism. This GI is physiologically opposite of GS, okay? So why we say physiologically is they're not directly competing with each other, but they're competing for the downstream effect of each other. So these are physiological antagonists. So alpha I is decreasing cyclic AMP, alpha S is increasing cyclic AMP. And again, that means that if you've got no alpha S activating ligands, you're going to decrease your baseline level of cyclic AMP. If you've got cyclic AMP activating ligands, then what's going to happen is this alpha I is going to inhibit that a bit so you can have a lower level of stimulation. So they're kind of, again, it's not black or white. It's like a little more, a little less kind of a thing. It's a dynamic process. Okay, so that's inhibition of cyclic AMP. Much shorter and simpler. You've just got the ligand binding to receptor, activating G protein, goes off and inhibits adenyl cyclase, and everybody's happy. At least I am. Okay, is everybody happy? Yeah, why not? Everybody's home. Nobody's wearing pants. Life is good. Okay, so what time is it? 10:10. Yeah, I guess, see, this next section of notes I threw in like 5 minutes before class because I'm like, oh my God, I don't have nearly enough material for today. So might as well go over it now. You know what? We're going to have to find a way to spend more time than just me lecturing because I'm going to get through everything I know really quickly. I don't know that much, frankly. So I've got a paper here that I want us to read. Maybe I don't have it here, but I'm going to post it up in Moodle. I'll talk more about this later, but I'd like you all to read this paper and then we'll talk about it probably next week to try and, help to the receptor pharmacology stuff to help to understand why it's important and how people use it to understand things. So I'll get a paper up probably today on that, and then we'll talk about it next week. But anyway, be that as it may, let's go into IP3 calcium. Okay, so this mechanism kind of revolves around a membrane phospholipid that gets so much discussion in cell biology. It's like so important. So you've got 4 membrane phospholipids, basically. You've got phosphotylethanolamine, which is, I forget how much, phosphotylcholine, which is a lot of the membrane, phosphotylserine, which is another one, and then a very low concentration of phosphatidylinositol. So your membrane, I believe, is about 5% phosphatidylinositol. So it's kind of rare, but it's there. And it's used for signaling an awful lot. So phosphatidylinositol is basically, here's your glycerol backbone, C. Glycerol is just 3-carbon chain with an alcohol on each one. And then you've got these fatty acids esterified to it, and your polar head group, that's a phospholipid. In this case, our phospholipid is inositol. So inositol is a 6-member ring, cyclohexane, I guess you would call it if it didn't have OHs on it, but here you've got an alcohol on each of those rings. So it's a sugar, basically. A sugar is a polyfunctional alcohol. So you've got a cyclohexane with an OH on every single carbon, and that's inositol. So that inositol then binds through a phosphate to this glycerol backbone. So that's a phosphodiester bond, just like the backbone of DNA, phosphodiester bonds. And let's see, once that is on there, like if these phosphates weren't here, okay, if these phosphates weren't here, it would just be PI, inositol phosphate. But then the inositol gets phosphorylated at the 5 position, okay, 1, 2, 3, 4, 5, the 5 position, and it also gets phosphorylated at the 4 position, 1, 2, 3, 4. So this is inositol 4,5 bisphosphate. Okay, inositol phosphatidylinositol 4,5 bisphosphate. Phosphatidylinositol 4,5 bisphosphate. Oh, it's right there. Holy shit. See, I'm trying to dig it out of my brain, which is full of KISS lyrics, and it's right there in front of me. Okay, so that's PIP2. Okay, PIP2. Now this is a big, ugly, negatively charged thing sticking out in the cell. It stands out like a sore thumb. And it can be used for a lot of things involved in cell division, but we're not going to worry about that here today. What happens is when you get activation of this enzyme, phospholipase C, it's going to break this bond, hydrolyze the bond, add water across it. And when that happens, you release this inositol with 3 phosphates, inositol trisphosphate, and diacylglycerol, glycerol with 2 big acyl groups on it. So what happens here, this phospholipase C gives us two second messengers, diacylglycerol, which is going to activate protein kinase C, and inositol trisphosphate, which is going to release calcium from the ER. Here's how that receptor system works. Very similar to cyclic AMP. You've got a receptor. In this case, let's make it a muscarinic receptor. Yeah. So we've got acetylcholine and a muscarinic receptor. It activates the GQ. Okay, this is now called GQ. So the alpha Q is a subtype of alpha subunit. So the GQ, alpha Q, activates phospholipase C. Phospholipase C cleaves this bond, and then IP3 goes in. Hang on. There's nothing a cat hates worse than a closed door. Oh. If I close the door, he'll want to come back out here. Okay. So that's the production of IP3. Why is that important? Okay, because I probably should have made another figure for this, but I want to emphasize here that this is your smooth ER, and your smooth endoplasmic reticulum is a store of calcium. It's a store of calcium because you've got this calcium ATPase here, circa, pumping calcium into it. And then you've got two different ways for it to get out. What we're talking about here is this guy over here, the IP3 receptor, which is a channel in the smooth ER. So when your phospholipase breaks down IP3, it then binds to that channel and opens it, and calcium comes out. Now I've got a better, I'm feeling distracted because I've got a better, got a much better figure for this. Now you're getting a peek behind the curtain, people. This is how the magic is done. So I've got a, you probably can't see this, and that's fine. But I've got figures. I've got a figure that I drew myself that I think is better than the one I put up there. Here we are. Oh baby, that is better. Good Lord, Dr. Slush, you are. I am very full of myself. Okay. So here is my figure. So what I like about this is here you can see here's your circa, your calcium ATPase, pumping calcium in here. This little guy, PL, phospholamban, we'll talk about him later in this class. So I didn't talk about that in cell biology. So let's not worry about it now. But here's your circa, pumping calcium in. You've also got mechanisms at the surface pumping calcium out. So you've got these huge calcium gradients, 2 millimolar calcium outside, 10 millimolar here in your smooth ER, 0.1 millimolar, that's 100 nanomolar, micromolar, I'm sorry, 100 micromolar. That's not right. No, it should be 0.1 micromolar. Yeah, it's very low inside, okay? So yeah, it's 0.1 micromolar, this is wrong. So anyway, you've got these huge gradients of calcium. And so your norepinephrine activating alpha adrenergic receptor activates GQ. GQ activates phospholipase C, breaking down PIP2 into IP3. IP3 then comes over and opens the IP3 receptor, and calcium comes out. Diacylglycerol, on the other hand, comes over and activates several different channels. These are receptor-operated channels. So when the protein kinase C phosphorylates them, calcium comes in through them. What I'm saying here is if the smooth muscle depolarizes, you also get calcium in through those channels, so I'm not going to worry about that. Okay, so protein kinase C then is causing calcium to come in, so you get an increase of calcium in the cell. Calcium then binds to calmodulin. Calmodulin is a regulatory protein. It's got 4 sites to bind calcium. When calcium is bound to calmodulin, it then binds to some target protein, usually a kinase. There are at least one other enzyme we'll talk about, but the calmodulin then will activate kinases, which then produce the effects that you see in the cell. Also, that protein kinase C will be phosphorylating some proteins, and that produces effects, in addition to phosphorylating these cell surface receptors and increasing calcium in the cell. So Bottom line being, all of this leads to increase of calcium. Calcium activates kinases through calmodulin, and then that phosphorylates proteins and produces the effects we see in the cell. Oop, and that's it. Okay, IP3 mechanism. Did I leave anything out? Active receptor, GQ subunit, diffuses away, alpha subunit, diffuses across the inside of the membrane, binds to phospholipase C, activates phospholipase C. Phospholipase C diffuses, breaks PIP2 into IP3 and DAG. PI3 binds to IP3 receptor, smooth ER. For calcium, bind to calmodulin, activates protein kinase C, increase in calcium, phosphorylates target proteins, blah, 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 blah. Okay. Reversal of this, when the hormone comes off the receptor goes inactive again. The alpha Q then hydrolyzes its GTP into GDP, shutting itself off. It then rebinds with the beta gamma subunit. The alpha Q then has come off phospholipase C, so it's turned off. IP3 gets broken down by phosphatases into inositol. It will eventually then be recycled back to become phosphatidylinositol again. But again, you've got those phosphatases that are sitting there waiting to take any phosphate off. So that's constantly going on, shutting things off. Calcium's pumped back into the ER. Calcium drops, calmodulin becomes inactive, and diacylglycerol is broken down by enzymes turning off protein kinase C. Okay. So that's about all I have for today. Any questions or comments? Okay, so I'm going to upload a paper. I must have it over there. I'm going to upload a paper for you today, and then I'll talk more about it on Friday, get you prepared for it, because there's some things you need to know before you can read the paper. And then I'll have you read it over the weekend, and next Tuesday we'll talk about it somehow. It's kind of hard. I've never done a paper. What I love to do in this class is we all sit together in a circle, and I tease everyone individually. Everybody gets teased. But more importantly, we get to sit and talk about papers and like, what does this experiment mean? What did we learn from this experiment? What did this show? That kind of stuff. Try and do it here on Zoom. I'm going to need a lot of class participation. Everybody's pretty quiet, unfortunately, but I need you guys to like step up on this one, okay? Otherwise, we're going to be done in like October, and I'm going to get fired because I won't be teaching my class after that. And I've got kids. They're like, what are they? I think there's a boy and a girl. But anyway, they need to be fed and everything. Okay, any questions, comments, discussion? All right, well, I'm going to let you go. I'm ending meeting in 3, 2, 1. See you Thursday.